The next type of transport that I want to talk about is endocytosis and exocytosis. That's where the cell membrane actually engulfs and takes in something. So if this is my cell, and here, I'll do it in another color, here's some starch, and starch wants to be taken in by this cell. The cell will actually surround and engulf it. So that would be like initial. Here would be my next cell diagram. And you'll notice how the membrane is literally going around and taking over. My last diagram, the contents are now inside of the cell, and that portion of the membrane right here that used to be part of the cell membrane has now formed a vesicle. So this process here where something is entering the cell is called endocytosis. There's two typical types of endocytosis that are discussed. One is phagocytosis. And phagocytosis usually is referred to as cell eating. It's usually solids. So potentially starch, something that doesn't dissolve well. Whereas the other type is called pinocytosis. Pinocytosis is usually taking in a liquid, dissolved solutes being surrounded and taken in. Now, as soon as that substance is inside of the cell, there is an organelle that would fuse here with it until it eventually became one large organelle together. And I'm hoping that you're saying to yourself, that would be a lysosome, because a lysosome contains all those hydrolytic enzymes to digest whatever just came into the cell. So a vesicle fuses with a lysosome. You'll also remember that we discussed the endomembrane system. That system is the system of membranes that are used to bring things into a cell or out of a cell. And here we're bringing something into a cell and surrounding it with a membrane. So that's why it's part of that endomembrane system. Now, if you look at the reverse, if I take an arrow this direction and this direction, what would be happening is something in a vesicle is being released out of the cell. That is called exocytosis. So exocytosis is the removal of something from a cell. If I could just refresh your memory, here is my cell, and here is the nucleus with that precious DNA inside. The RNA leaves the nucleus and goes to a ribosome. Here's a ribosome. The ribosome might be part of a series of ER, endoplasmic reticulum. That ribosome is capable of using the RNA from the nucleus and building a protein. So the green is going to represent this protein that I've built. The protein can be transported by the system of tunnels, this network of the ER, until eventually it makes it to the Golgi bodies. Golgi bodies, I usually say they look like a stack of pancakes here. The Golgi bodies package proteins into vesicles. So here's that protein traveling through until eventually it is enclosed within a vesicle. That vesicle can then fuse with the membrane and be released outside of the cell. So again, that's also all part of the endomembrane system. Membranes carrying a product until it is out of the cell. So that was our nucleus. This is our endoplasmic reticulum. That is a ribosome. These are Golgi bodies, and these are vesicles, and of course that is the cell membrane.